Metabolic therapies, such as nutritional ketosis, have been proven effective for seizure disorders and also for several neurological disorders. So today I'm going to talk about some recent studies in animal models that focus on the enhancement of the SZENT-GYÖRGYI-KREBS cycle. In the healthy brain, glucose is the primary metabolic fuel, but in neurodegenerative diseases, there is impairment in glucose transport and metabolism, and there is a mitochondrial dysfunction that leads to energy deficit. These features are all pathological hallmarks for Alzheimer's disease or Parkinson's disease, seizure disorders, or traumatic brain injury. However, ketone bodies and TCA cycle intermediates function as alternative fuels and when glucose is not available. In fact, as you heard before, the brain is able to utilize energy, 60% of its energy from ketones when the glucose availability is limited. So our body is able to utilize energy from free fatty acids when the hepatic ketogenesis converts the free fatty acids into ketone bodies, such as beta-hydroxybutyrate, acetoacetate, and acetone. By this mechanism, the ketone bodies accumulate in the blood, and this state of normal physiological ketosis can be therapeutic. During starvation, the blood ketone levels become elevated, and this is an essential survival mechanism that ensures metabolic flexibility. The unpleasant side effect is hunger. The therapeutic ketosis can also be achieved by a high-fat, low-carbohydrate ketogenic diet, which can only induce a modest elevation of blood ketone levels, and it requires a very strict dietary restrictions. MCT oils or medium chain triglyceride can only induce a modest elevation of blood ketone levels, and they are often associated with GI disturbances. Exogenous ketone supplementation, such as ketone esters, would circumvent such strict dietary restrictions. For this reason, ketone supplementation and other forms of energy intermediates are being developed and tested as metabolic therapies in our lab. Esters of beta-hydroxybutyrate and acetoacetate can effectively induce rapid and sustained elevation of the ketone levels. So Dr. D'Agostino and his chemist colleague synthesized a ketone ester, a 1,3-butanediol acetoacetate diester, which I'm going to just mention as a ketone ester. Presently, the ketogenic diet is the most efficient metabolic, non-pharmacological approach for treating epileptic patients who don't respond to drugs. The diet can successfully work in about 50% of the patients by eliminating the seizures, and in the remaining 50%, the quality of life improves. The anticonvulsant mechanism of the ketogenic diet correlates with the blood ketone level elevation, especially the acetoacetate and acetone. So to study the anticonvulsant effect of the ketone supplement, we used the CNS oxygen toxicity model. Breathing 100% oxygen at the barometric pressure of 2.4 atmospheres absolute or higher increases the risk for seizures. This problem is a limiting factor for Navy SEAL divers and also a limitation for patients who are using hypobaric oxygen therapy. Since the hyperbaric oxygen provides a unique, quickly reversible, and highly reproducible stimulus for seizure in animal models, therefore we consider it as an effective model for assessing anti-seizure strategies. So we use this seizure model. We dive rats in this hyperbaric chamber that we previously implant with telemetry devices that can measure EEG, ECG, and EMG. So we put the rat in the chamber. We pressurize the chamber to 5 atmosphere absolute with 100% oxygen. And we can detect the seizure based on the behavior of the animal that we follow on live video camera. And we can also see the EEG signal. And when we detect the seizure, we quickly flush the chamber with air, which immediately stops the seizures. And then we decompress the chamber. 30 minutes before the dive, we give either water or ketone supplements to the rats, either butanediol or the butanediol acidoacetate diester, the ketone ester. And these graphs are just examples where you can see how do we detect the seizures based on the EEG signals. We demonstrated that a single oral administration of the ketone ester caused rapid and sustained elevation of the beta-hydroxybutyrate, acidoacetate, and acetone levels in the blood, and also increased the latency to seizures by more than 5 times. Instead of a single oral administration, we next fed the rats for 28 days with the ketone ester supplement and also with another ketone ester supplement that we called ketone salt, ketone mineral salt. And we analyzed the changes in the metabolomic profile. We found several-fold elevation of blood and brain ketones in treatment groups versus the control. We also found several-fold elevation in the Krebs cycle intermediate in the treatment groups relative to the standard diet groups. So ketones may have a therapeutic effect against neurodegenerative diseases that are characterized by impaired glucose metabolism. Currently, the most prevalent motor neuron disease is ALS, and no cure or treatment is available. In order to understand the mechanism of the disease and to test different therapeutic approaches, we use a transgenic ALS mice model. The most commonly used ALS transgenic mice model is the SOD1G34A model. And in this model, the mice start showing symptoms around 100 days of age when the legs become weaker, they have difficulty walking, and eventually the hind legs become paralyzed, and in a couple days they die. Zhao and colleagues reported functional improvements on the Rodarod test and also histological improvements when the transgenic mice were fed with a ketogenic diet. A few years later, the same group administered caprylic triglyceride, which is a medium-chain triglyceride, to the transgenic mice model, and they found, again, motor function improvement tested on the Rodarod test, and also they found reduced that of motor neurons in the spinal cord when the mice were fed with caprylic triglyceride. The survival time, however, didn't increase in either of these studies. Anecdotal reports from ALS patients describe symptomatic improvements in motor function following the administration of the supplement complex called the Diana protocol. The main ingredient of the Diana protocol is arginine alpha-ketoglutarate. in combination with ATP production supporting agents. The DNA protocol was developed to slow the progression of the disease, which is caused by the premature death of motor neurons, by supplying alternative fuels for the neurons and the glial cells. The DNA protocol supplementation, when added to the standard rodent diet, improved motor function that was tested on accelerating rod, grip test, and hanging wire test. And it also delayed the progression of the disease. The neurological score was significantly better in groups that were consuming the standard diet with the DNA protocol between age 116 and 135. In addition, the treatment significantly extended the survival. 72% of the animals in the standard diet plus the DNA protocol group lived past 125 days, while only 9% of the animals in the control group lived past 125 days. Since the ketogenic diet had been proven to improve metabolic efficiency in seizure disorders, it was suggested that the diet can be useful for neurological disorders such as Alzheimer's disease, especially since many Alzheimer's patients experience seizure. In Alzheimer's patients, similarly to other dementia, there is a hypometabolism in the brain. There is an energy imbalance in several brain areas, especially in the hippocampus. It was suggested that providing ketone bodies as alternative fuels for the neurons, it can bypass the metabolic deficits in the Alzheimer's brain. 2 transgenic mice models were tested when administering the ketogenic diet. 5 months old amyloid depositing mice, transgenic for mutant amyloid precursor protein and presenilin-1, APP-PS1, and tau depositing mice, transgenic for tau, were fed with a low carbohydrate, medium chain triglyceride-rich diet, which induced ketosis. Motor performance was assessed by accelerating and non-accelerating rod. Both the genotype and the diet had an effect on their motor performance. The average latency to fall was significantly greater in mice that were fed with ketogenic diet in both accelerating rod and also in non-accelerating rod experiment. So in conclusion, we saw that ketone bodies serve as alternative fuels for the cells. Elevated blood ketone levels improved motor function in several animal models. We saw reduced death of motor neurons in the ALS mice model. And elevated blood ketone levels also led to elevated TCA cycle intermediates. And we saw an increased survival in the ALS mouse model. In summary, we can say that the ketogenesis produces a metabolic profile that is consistent with increased metabolic resilience. And with that, I would like to thank our lab, Dr. D'Agostino's lab and Dr. J. Dean's lab at the University of South Florida, and also I would like to thank our funding agencies, and thank you for your attention.